We start again, and thank you for listening to me. I have the utmost pleasure to introduce the undergraduate who has worked with me for the past 3.5, almost 4 years, and that is Devon Colby. Devon has worked in Dr. Jibo Yang's lab, which is an analytical chemistry lab, and he has done some wonderful work with the development of a new mass spectrometry technique. And I will let Devon talk more about it.
Okay, hello everyone. I'm Devon Colby. Apologies in advance if I have some technical difficulties, so Wi-Fi here isn't the best. And I'd also like to speak to Rama's last point in her slides there. I have encountered a number of failures through these. But so today I will be presenting to you over my latest research project, which is developing a method for the ambient mass spectrometry imaging of samples with topographical variation using the single probe. Now, this research is important as mass spectrometry imaging has a variety of uses, such as through environmental research, forensics, or medical applications, among others. For instance, it can be used on tissue biopsies, finding molecular distributions for analytes such as drug metabolites, or can be used to help classify patients for aiding therapy decisions through detecting tumor cells and their surrounding environments. Now, just a bit of background to start off here. Mass spectrometry is a sensitive analytical technique which detects molecules at even pico and femtomolar concentrations and is generally used for gaseous and solution-based samples. Within mass spectrometry, there are both non-ambient and ambient techniques. Non-ambient generally occur or always occur under vacuum conditions and are generally less conducive to biological sample analysis as they require a lot of sample pre-treatment and can tend to hurt the integrity of these samples. However, ambient techniques, which occur in open environments, can be better for the analysis of biological samples in some cases, depending on what you're looking for, as they require less sample pretreatment usually and can be used for so they are more conducive. Now, mass spectrometry imaging is a subfield of mass spectrometry and is a molecular imaging method which is used to visualize the spatial distribution and relative abundance of analytes, a variety of analytes actually, over a sample surface. Now, a single probe is a multifunctional device created and used by the Yang Lab and is capable of performing ambient single cell mass spectrometry and mass spectrometry imaging. The single probe MSI method, as visualized in the figure to the left, where you can see here, works through scanning over the sample surface and extracting molecules through microscale liquid extractions, then directly injecting these molecules into a mass spectrometer for analysis. Now, within mass spectrometry, the majority of studies occur with flat samples. This is because generally working with uneven or non-flat samples tends to lower the spatial resolution of the resulting mass spec image. However, there is an interest in the analysis of non-flat samples. Currently, the single probe method, much like these other methods, is limited to the analysis of flat samples. And in the single probe's case, this is largely due to unevenness in sample surfaces, causing the single probe to either clog or break, in which case it will lead to lower spatial resolution or just experimental failure. Towards this, my project aims to adapt the single probe MSI method towards use on samples with topographical variation in their surfaces. And this project has both a hardware and a software component. I'll start with the hardware part, in which I have developed a 3D printed platform seen to the right, which allows the single probe to adjust for variations in the topography of the sample. And as you can see here, these capillary tubes act as springs to allow the allow the probe to adjust its position relative to the sample such that it won't break or clog when moving along it. Along with the hardware component, there is a new program being developed to help deal with uneven surfaces. This program will be used to control the movement of the sample stage during experiments. Now, within this project, there are two sample surfaces which will be used. Those are smooth and rough. With smooth samples, the current program will be used. Now, these samples will generally be in the form of something such as a glass test tube, whereas rough samples will mostly be biological and will require the use of the new program to avoid damaging the probe. Now, within these methods, as I said, there will be two different surface-specific programs, and I'll describe them here. You can see the current program schematic on the top and the new program schematic on the bottom. I will go over those. Now, within the current program, there are multiple lines within each sample. Within each line, the sample stage will move in the X direction relative to the single probe, scanning as it goes. Upon completing each line, the sample stage then moves away from the probe in the vertical direction as seen here to the dwell position. And it will stay there for a predetermined amount of time known as the dwell time. Following the dwell time, the sample stage then moves to the next line, including in the vertical direction, and begins again, repeating this process until data for all lines has been acquired. Now, for the new program, samples will still have multiple lines, and the probe position will still remain constant through data acquisition. However, the sample stage will return to the dwell position following each individual scan before returning the sample surface to the probe at the next scanning coordinate. This is according to the current plan for the program being made. And so this motion will give a tapping-like motion to the sample stage, which will then allow the probe to continue to account for sudden changes in the sample topography and to not catch on any roughness within samples. Along with this, it will still, and as you can see here, it will, you have the sample here, you have the sample on the top here, that is where the data is being acquired, and you have the sample position here, that is at the top of the tapping motion, in which case it will be alternating between these throughout data acquisition, following which it will still go to the dwell time, and this will repeat again. Thus far within this, proof of concept data has been acquired for the newly designed platform. Ibuprofen and rhodamine, among other analytes, have been used and applied to the test tubes via fingerprints. Distributions of these analytes have been mapped then, and an example of this can be seen to the right in this image. This is about a 1 millimeter square sample area, and you can see this here, and these are the aspects which it picked up via the distribution and relative abundance of ibuprofen in the sample area. Now, in the future work, I will improve the performance of the platform to enhance ion signal stability and the spatial resolution of the MS image. Along with this, the platform will be used to analyze larger sample areas And this is because, as you can see, the sample area here is much smaller than the actual size of the fingerprint. So we want to pick up more aspects of the fingerprint to just have a better view of it. Along with this, the new program will be finished for use on rough samples, and then this will all be combined for the adapted method, which will be applied towards uses such as the analysis of drug diffusion rates and depths within tissue samples, among other applications. I would like to give special thanks to the members of my group, specifically Dr. Jibo Yang and Xiang Tian, for helping me through this project, as well as Dr. Kothapalli for helping me throughout my entire time in research, including here, especially in forming collaborations, which I am currently using to help formulate this new program. And here are my references. And thank you. Are there any questions?
All right, wonderful. Thank you very much. I'll get you to leave the presentation up for just a second.
Oh, sorry.
Yeah, that's all right. Go ahead and share it back again, because I think it might be helpful for at least a couple of questions.
Yeah, I acted too soon.
There you go. All right. So the first I had a couple of them along the way. So the first is what kind of resolution can you achieve with this imaging?
See, so with the current method, I don't remember the exact numbers for it, but I think it was around 20 microns from what I remember. I could be remembering that wrong, though. It's been a little bit since I saw that figure.
Okay. I mean, that's pretty good. Okay. And I hope I understood the technique properly. So you have basically you have a solvent that comes down, hits a surface, and picks up something on that surface, and then carries it back up to be analyzed. Is that the basic idea?
Yeah. So there is a liquid junction which forms at the tip of the probe.
Okay.
And when that hits the analyte, it is able to pretty much, I guess you could say, dissolve the analyte. And there is a current going through this, which ionizes it for the mass spec, for use in the mass spec.
Oh, okay, I got it.
Yeah,
okay.
Sorry, I forgot to add that part.
No, that's fine. This, like I said, this is an interesting technique because I'm familiar with mass spec and even a little bit of mass spec imagery, but this is a new one to me. So this is really I'm interested in it. So
yeah, this
what?
one's an ESI. It's an ESI emitter over here.
Okay. What kind of solvents can you use? Or I'm assuming like you can use water, maybe an alcohol or something like that. But
so from what I remember doing the experiments, I remember using generally acetonitrile and I think formaldehyde as part. It's a mix.
Okay.
That I was using from it.
Do you anticipate? I mean, this may be outside of the work that you've done, but do you anticipate that? Like, let's say you're looking, like you're probing for a specific analyte. Could you alter the solvent to sort of, improve the signal from that analyte? Like, if say it's more soluble in a particular solvent, could you sort of try and target it?
Yeah, I believe I can. That to a degree that would help. I know from what I remember, Dr. Tien now. He, I think from what he remember, a large part of what he used was methanol within his solvent. So that might help with certain analytes as well.
Interesting. Okay. And then somebody had a follow up on the resolution question that I had asked. Arctech wanted to know: have you thought about using piezoelectric materials to control the probe? And if so, do you think that would provide better or worse control over the probe position?
So I guess I can look into that. I can't say I know much about piezoelectric materials.
Okay.
So that is something to consider in the future then.
Okay.
Or to look into.
Yeah, that's interesting. I worked a little bit with a scanning electrochemical microscopy setup, and it used piezoelectrics to do something. Not obviously not the exact same thing, but it's a similar idea where you have a probe that's coming into close contact with or into near contact with a conductive surface, and you use a piezoelectric to move in the X, Y, and Z. And so the architects that is a good idea because it may provide you a little bit finer control um, over the movement of the probe itself. What kind of
Oh,
time does
and
it take?
for
Okay.
clarification on that, the
Hmm.
probe itself is actually in a constant position.
Okay.
The sample stage is what is being moved.
Okay. All right.
Yeah. So I will bring it to here.
Hmm.
So you can see the probe here,
Yep.
or kind of.
Yeah.
It's somewhat hard to see,
Sure.
but so let's say that it was positioned here. The sample stage would be directly under this,
Okay.
and it would be moving in relation to the
Okay.
probe.
I got you. So the probe is fixed, and then you achieve the movement by moving the sample around underneath.
Yes.
Okay. I just had a question and it just ran away from me. Shoot.
My apologies. That might
No,
have
no,
been
no. That's fine. I don't remember what it was. Um. Yeah, it's gone. Yeah, it's completely left me. Um. This is like I said. This is really neat. I had never heard of doing this, but it's a really the way you've explained it. I mean, it's a really interesting way to do it. Um. So what kind of what are the applications for this? Like let's say 10 years from now, if everything continues to progress, what's sort of the vision for how this would be used?
So in the past, I know that some of these things, some I don't know all the applications. Obviously, I
Hmm.
know that some mass spec imaging has been used on surfaces such as biofilms, though I don't know how much that would be useful with this, just because I don't know how much variation there is in the surface. But
Hmm. Sure.
but I know that in terms of like tissue samples, those can sometimes have a deal of an amount of variation within the topography of the sample, and so that can wreak havoc with it if it isn't dealt with in preparation.
Okay.
This would eliminate some of that need for preparation, and even I guess it could possibly be prepared incorrectly and still cause problems.
Oh, absolutely. Yeah,
Uh,
absolutely.
and so then this would be great for tissue sampling, and as well as there are certain forensic applications of this as well.
Okay.
Like for the tissue sampling, one thing you can look at is like you can look at kidney biopsies,
Hmm.
and you can see something such as a drug diffusion rate into the kidney.
Oh, okay. That's really
And
cool.
yeah.
It's really cool. Okay, I actually did remember the question that I was going to ask you a minute ago. How long does it take to do this imaging analysis, or like the
Ooh.
figure that you showed of the thumbprint? How long does that take to collect?
Okay, so that 1 millimeter square area,
Hmm.
that took about 4 hours.
That's not terrible. I mean, given
Yeah.
that you're working with mass spec, that's really not that bad.
Yeah, it's I did about 50 lines on that one,
Oh man.
and so that I mean, it felt like it took a while,
Right.
though I also left for part of it.
Yeah, yeah. Always good when you can do that.
Yeah, there's not much I can do once it starts,
Yeah.
but then it's gonna take significantly longer, though we're using an orbit trap right now,
Hmm.
and we're gonna switch to using an ion trap so that we can do faster scanning there
Okay.
and allow the probe to do a larger surface while still maintaining a fair amount of signal stability.
Okay.
Yeah,
Do
because
you
that's one of the problems that we found is
yeah.
that it.
Oh, that's a good point, Rama. Here, I'll tell you what, Rama. I will unmute you. Uh, oh, maybe. All right, it's not letting me. So Rama made a good point that you make the probe yourself. Um, and so how do you make those?
Okay, so I'll go back to that part. Is it specifically for this probe right here? I'm guessing. So
Okay.
here, what we do is we have some dual bore glass tubing here,
Okay.
or quartz tubing, my bad.
Mhm.
And we have these thin capillary tubes, which you can see here as
Okay.
well. And we insert these into there after pulling the dual bore glass quartz tubing
Yeah.
in a glass puller, or I don't exactly remember the name of the equipment.
Yeah. Oh, is it a? Yeah, I know what you're talking about, though.
Yeah.
Yeah.
But yeah, it pulls the glass. We snip the ends of the
Mhm.
tip here. That mine it varies. I tried to get around 10, but during the experiments I kind of have to snip it every 10 microns, I mean.
Mhm.
But I kind of have to snip it more quite a bit of the time, just because it still can tend to clog, because some of the
Sure.
analytes or such can be a bit
Yeah.
annoying.
Yeah.
Yeah. And then we use UV glue to set these capillary tubes in here. Now, usually, as you see here, this would be set on a glass slide,
Okay.
but mine here, once I've set the emitter here, and if you can see, there's a capillary coming up here. That is the solvent providing capillary.
Okay.
And once I've set those in using the UV glue here,
Okay.
I will then attach this. I will put this through the probe hole,
Mhm.
and I will attach these capillary tubes here to both the capillary walls and the probe itself, the base of it,
Mhm.
using the UV glue as well. Same with the emitter here.
Okay. Wow.
That part's quite time consuming.
Yeah, I was about to say I don't I feel like my sausage hands would not be able to do that very well. So I'm duly impressed at your synthesis.
It can be very aggravating, actually.
Yeah, no, I imagine. I was just saying in the chat, I used to make ultra micro electrodes, and so it's a similar idea where you pull a piece of glass tubing, get a very fine point, and then you have to work a wire down in there and polish it down and get all that fun stuff. But that's yeah. So just making the probe itself is very impressive.
They fail a lot, actually.
Yeah. Yeah. So how do you average, like when you do the imaging scan that you had, do you go over the same area multiple times, or is it like a once through, or?
So, I wish I had an image of that here. I'll make sure to put that in my next time I make one of these.
Okay.
But I guess I'll have to go with this.
Mm-hmm.
Here you'll see. So this area here gets divided up into 50 lines.
Okay.
Each one is about, I guess, 2 microns. I'm a bit tired right now, so
Yeah, I think, like I said, that would be the 20 microns that you were talking about. So that makes sense.
Yeah, and yeah, 20. My bad.
Oh yeah, no worries.
Math late at night is hard.
Yeah, very much so.
But yeah, so they're each about 20 microns apart, and then after each line, the program will move it in both the Y and the Y, Z, and X direction towards to the beginning of the next line.
Mm-hmm.
And so you don't usually go over part of it again, though, because of how the solvent interacts with the glass and such,
mm-hmm.
it can sometimes pick up things from nearby lines, which can cause problems.
Okay. So I guess it's not a destructive technique, but it does change your surface a little bit by the fact that it's having to pick up the analyte. Is that a reasonable way to put it?
Yeah, it can also cause some minor scratching of the surface as well, just because you're running a sharp object over it. So.
Mm-hmm. Okay, that makes sense. That's a really, like I said, that's a fascinating technique. I'll be sure to keep an eye out for this, because this is a new one to me. So that's very cool. All right, so I'll give the chat, if there's any other questions, go ahead and get them in. We'll wait a little bit, because there's usually a little bit of delay between the between my talking and the actual response from chat. But this is really cool.
Okay.
And so you're looking for an MD PhD program now?
Yeah. That for that part, I'll be aiming for more of an immunology side, but I can still be developing technology and therapeutic methods for it.
Okay,
Or
very
at least
cool.
that's the goal.
Yeah, absolutely. That's a good goal. You can always use more of that. All right, I think that's going to be all the questions. And so I will thank both Devon and Rama for coming on. Like I said, I really appreciate you guys doing that. Both talks were excellent, just as I was hoping, and I really appreciate you guys sharing your work.
Thanks for having us.
Yeah, absolutely. And so with that, thanks everybody for coming out. If you, like I said, these talks will go up on YouTube in a little while. It usually takes a couple hours to process, but both of them will be up there, including the Q&A after each one. If you're new to the chat and you would like to join our Discord, I'll throw the link for that in there into the chat. It's a friendly Discord. We have places for collaborations, manuscript help, all that good stuff. And yeah, I think that's going to be it for tonight. So again, thank you to Devon and Rama. And with that, I think we will call it a night. And we'll see you all next week. Have a good night, everybody.